Thank you very much. It's a real pleasure to be here in beautiful Berlin, presenting our framework to read and write organs using genomics and computational tools. And I will be telling you how we apply this framework in the context of female reproductive tissues. And you may be sitting and thinking why I have chosen the female reproductive tissues for this talk. So first of all, we are all here thanks to the reproductive tissues of our parents. So the reproductive system is the one that ensures that the genetic material is passed from generation to generation, and in this way perpetuating life. And yet still how the reproductive tissues develop and regenerate is a black box in humans. And there are multiple reasons of why this is happening. So first, it's difficult of accessing human tissues. Second, reproductive biology is still under research. There are 4.5 more papers in non-reproductive tissues than in reproductive tissues if you have a search on PubMed. And also, there is a funding gap on women's health, which was actually highlighted by Nature a few weeks ago, and that includes all the study in reproductive tissues. Also, there are huge differences between humans and mice, including physiology of the system, the hormones, and functional differences, and this makes it difficult to translate information from mouse to humans. And then there is still a lack of complex in vitro models that really recapitulate the complexity of these models or this system. And there are more and more systems emerging, like organoids, which are epithelial, also co-cultures with stromal cells, but still there is still not developed this complexity in a dish. So there is a long way towards this. So all of this has consequences for reproductive conditions, which are actually widespread. And I will just give some numbers. So for example, endometriosis affects 10% of women in reproductive age. Or then, for example, pregnancy complications account for 8% of all pregnancies, or even some cancers, such as ovarian cancer, which accounts for 2.5% of cancers in women, and it is one of the most deadly cancers in women. So altogether, all of this has a significant morbidity in women and an increase on infertility. And we have now a unique opportunity on change these numbers by really investing in knowing more about the physiology of the system that will also allow us to develop better and more accurate and sophisticated in vitro models. So how we on our team do that, learn about the physiology and think how can we improve this complexity of the models. So to tell you more about it, let me go more into the human body. Let me take a deep dive into the human body that, as you know, is formed by different systems, which include the reproductive system. And then if we zoom into the reproductive system, we have the different organs, which in the case of the female reproductive tissues includes the ovary, the fallopian tubes, the uterus, the cervix. And if we continue to digging into the tissue, then we go into specialized structures, for example, the ovarian cortex that contains the eggs. And finally, we arrive to the cells, which are the fundamental units of life. So these different cells in the tissue arrange and to form like this specialized niche on functions. And to really understand the function and the functionality and physiology of the tissue, we need to go into what are the cells that are forming the tissue. So all cells in the body, or the majority of cells in the body share the same genome, and yet we have cells that have different shape, different function, and different sizes. And what is creating this diversity? So what is creating this diversity is the genes that are expressed within a cell that is known as the transcriptome, that will determine the proteins that are expressed and the function of that cell. And what happens during development is that we have regulatory circuits that control what of those genes are expressed, and that gives identity to the different cells, which then self-organize into tissues, creating even more diversity, and finally creating the different systems that we have in the human body. So to know more about the cells and the function of the cells, we need to look, or we can look at the transcriptome, so the genes that are expressed. And there is a technology that allows us to do that is called single-cell transcriptomics, and that I will be using during my talk. And basically, in this technology, we have the tissues where we then need to isolate the cells, and we can also isolate the nuclei. And then from those specific cells, we isolate the RNA, and then we sequence, and then we can get all the information for the genes that are active within a cell. And that means that if we do this at scale, then we can quantify all the diversity of cells that are present within a tissue. And this is the foundation, or these technologies that what led to the development of the Human Cell Atlas initiative, which is an international initiative that I am part of, that aims to map all cells in the human body. And it's an open initiative that you can all join at humancellatlas.org, where you can look at the different cells and the genes that are expressed in the different organs, and they are distributed in bionetworks, and which include the different tissues, but also the organoids, to understand the heterogeneity of the organoids. And in particular, the work that I will be presenting is the work that my team did as part of the reproductive tissue bionetwork. Okay, so far I have been talking about the single-cell technologies that if we make an analogy to this fruit salad, what it will allow us to quantify is the different fruits that are part of this fruit salad. And this is great, but in addition to that, we want to know how these fruits are organized in this fruit cake. So what fruit is close to each other, what are the specific structures that are forming? Or in other words, how cells rearrange into tissues and then form functional structures, including the uterus, the brain, the gut. And for this, there is another technology that I want to introduce that is called spatial transcriptomics. So in spatial transcriptomics, what we do is to measure the transcriptome directly on tissue slides. And for example, in this 10x Visium, basically what we put is the tissue directly on a tissue slide, so then the RNA will diffuse, and these slides have a spatial barcode, so they have these barcodes with these spatial barcodes that allow us to resolve the transcriptome in a specific voxel together with where are they located, so we can know about the identity of the cell and also the spatial localization. There are other technologies that are more image-based, such as Xenium, in which basically the advantage of this is that we know the boundaries of a cell, so we can quantify the specific RNA that is present within a cell. And Yeah, and the disadvantage of this is that we need prior information about um, the, the, the tissue that we want to profile, because we are limited to a specific number of genes that we are screening. For example, in this case, it's 300 genes. Okay, good. So far, we have seen that with all the single cell and these special transcriptomics, we can build all these cell atlases, and this contains a lot of information, which sometimes can be a bit overwhelming. And the key here is to be able to ask the right questions to the data. And to do that, we need help of the computational and machine learning tools that we and others develop. So if we use these tools, then we can ask questions of what are the cell states that are present, what genes programs are active, what is the location of a cell into the tissue, or what are the signals that are present. And of course, you can do this in different combinations, such as, for example, comparing disease and healthy, or in vivo and in vitro. And I will go a bit more into detail for what are the signals that are present, because this is something that my team is hugely interested. So cells are not alone in the tissue, they are close by to others, and they communicate constantly. And a way to communicate is through ligand receptors, where we have the ligand, that is a protein that is expressed by a cell, which then binds to a receptor, which is the receiver, that is a protein that is attached to the molecule, to the membrane of the receiving cell, and upon the binding of the ligand triggers a specific signal, which ends up with the transcription factor being active in the cell. And this is important because the transcription factors are the ones that will regulate the expression of the genes in the cell. And indeed, the transcription factor, to do their action, they bind to specific regions in the genome, and because they are so important to determine the expression of the genes into a cell, there are multiple technologies on how you can measure if they are active or not. And I will introduce briefly the one that we use, that is single cell attack sequencing, in which you are measuring the regions of the genome that are open, and using computational tools we can infer what of these transcription factors are binding to these open chromatin regions. Okay, so during my postdoc, I developed the first tool to infer cell-cell communication from single-cell genomics and data. And since then, my team has been improving the tool much further to be more precise and predictive. And basically, the basis for this tool is very simple. So we measure the expression of ligands and receptors on the different single-cell data set, and then we look at the transcription factors, and we use them as a sensor that these ligand receptor interactions is happening in the environment. And to do that, the tool consists in two modules. So first is a database of curated interactions of ligand receptors and also downstream signal. And the uniqueness of this database is that we also take into account the protein structures and the complex that the proteins form. And then there is a method in which, as I was telling you, we look at for ligand receptor expression in the different cells, and then we also measure the transcription factors, and then we use them as sensors that this communication is happening. And then also we use the special transcriptomics to filter and to get on these interactions that happens into a particular cellular niche between cells that are relatively close. Okay, so with all of these single cell and special transcriptomics, what we are doing is modeling the in vivo data. So as I told you before, the single cell genomics data allow us to gain information about the identity of cells that we have. And then we have a special transcriptomics that allow us to put together these cells into a context and to look for the tissue architecture. And here I want to introduce a new concept on how we can utilize all these amazing data to then benchmark our models, our in vitro models, and also learn from all this in vivo data to even improve our models. And this is what we call the in vitro modeling and the start writing the organs, in which we can look for a specific differentiation process where we have a stepwise differentiation process, and we can guide this by getting all the information from the single cell data, because we have the identities but also the regulatory pathways that are happening. And likewise, when we start putting more complex systems, then we can get all this information from all the spatial atlases, knowing which cell is close to each other, but also starting to learn gradients about the specific pathways that are, or specific molecules that are happening into the tissue. So making use of an analogy, that will be like building Captain Jack, in which we have the pieces, because those are all the single cell data that we have, but we also have the instructions from all the in vivo data, which will be the cell-cell communication, prediction, all the pathways and the transcription factors. And with all this information, we can reconstruct Captain Jack, or in other words, start benchmarking and improving the current in vitro models. And in my team, we have been utilizing this framework in different contexts, mainly in the area of immunology and also reproductive tissues. And in the case of the reproductive tissues, we have been working in different systems, in vivo and in vitro, across lifespan. But for today's presentation, I'm just going to focus on our work in the uterus, before and after, during pregnancy. So for this, let me introduce the endometrium, which is the mucosal layer of the uterus. And as you can appreciate, it's an epithelial tissue which is formed by the lumen and the glands. And basically, the glands contains the secretions that will enable the embryo to survive during the early stages of pregnancy. And then the lumen contains the cilia that will spread these secretions to the uterine space, and also that will allow the interactions with the embryo and enable the implantation. So what is fascinating about the endometrium is that it regenerates cyclically in response to ovarian hormones, the estrogen and progesterone, and this really is a perfect regeneration that occurs without scarring. So there is a lot to be learned from this tissue. And it's quite fascinating that this happens more than 400 cycles over a women's lifetime. So it goes into cycles, as I'm saying, and then we have, during menstruation, there is the shedding of the tissue, and then during the proliferative stage, in the presence of estrogen, cells regenerate, and then there is a differentiation in the presence of progesterone that is happening in the secretory phase. And what we asked in this first work was, what is the composition and the interactions that are happening in the adult endometrium that is enabling this perfect regeneration to happen in healthy, because we just wanted to have a reference before we start understanding disease. And also we wanted to understand what are we recapitulating in our in vitro models, because in this particular case, this cycle is quite different from what happens in mice models. We really need the human models to study the endometrium. Okay, so we first start with the single cell data, and all the data that I'm presenting here is from our published paper. And here basically we have single cell transcriptomics data, and in this data each dot of the plot represents a cells and cells cluster based on similarities on the transcriptome. If we look at the proliferative stage of the epithelial cells, we have two epithelial cells that are distinguished by the expression of the LGR5, and then we wanted to know a bit more about those and where they sit in tissue, and that's where we use the spatial transcriptomics tool, and then we can find that these novel populations that we define, one is in the lumen and the other in the basal layer of the glands, and with this we have this partial model where during the proliferative phase we have these two epithelial cells that are occupying different niches in the tissue. So that's great, but then we wanted to know how those give rise to the heterogeneity of epithelial populations in the secretory phase that will be the ones to do the function. And again, we see the LTR5 positive and negative, but also the ciliated popping up. And again, if we put this into the context of a spatial transcriptomics, we can see again the location of LTR5 positive in the lumen and LTR5 negative in the base, in the glands, and again having this model where now we uncover the heterogeneity of the epithelial cells across the cycle. And that's great, but what we really want to understand is what are the signals that are pushing this identity of the epithelial cells. And to do that, we took advantage again of the spatial transcriptomics, and basically we cluster the cells based only on information of the spatial location. And what is very cool about this system is that the clusters that they form are associated with the identity of the cells, but also with the spatial needs they occupy, meaning that the spatial or the tissue architecture is quite important for the identity of the cells. And when we look at the pathways, we can see the cell, like the regions in yellow that correspond to the lumen have expression of Wnt activatory signals, while the glands in blue have expression of NOT activatory signals. And indeed, we use better resolved imaging methods to look at these gradients, and here is just an example of NOT and how when we go deeper from the lumen into the tissue, we have higher expression of this molecule. And we did that for the others as well and quantify the signal. Okay, then we wanted to know what are the cells from the environment of the endometrium that are giving this signal. And for this, we use CellPhoneDB. And the way to look at these plots is that we have the receptors, the ligands on the left, and then the expression of the receptors on the right. And then you can see the arrows, how they go from one to another, and then we have the different cells. And what we can see here is this expression of DKK1, which is a potent Wnt inhibitor, which is expressed by the fibroblasts of the endometrium, which are called stromal, and the ones that are just in the secretory phase. And you can see how this signal is expressed surrounding the glands. So with all of this, we have a model where not only we uncover the different identity of cells, but also the different signals, including this disbalance between Wnt and NOT in the different tissue microenvironments. And that's great, and then we ask, can we use all these reference to benchmark the in vitro models that we are using in the lab, and even to play a bit with them and try to inform if we can alter the different lineages that we get in culture by altering the different environments that we are recapitulating by gaining information of this in vivo data. So to do that, we use organoids, and in particular this experiment of the organoid was done by Margarita Turco, who was in Cambridge University by then. And basically, we grow, in this model, you grow the organoids, and then you expose them to hormones. And then we profile them by single-cell transcriptomics. And in the expansion media, you don't have a lot of heterogeneity, but as you put estrogen, then you can see popping up populations that respond to estrogen, and also some ciliated populations. And in the presence of progesterone, we have more the secretory populations popping up. And I want to spend a bit of time on saying how we can do that in a quantitative manner and how single cells allow us to do that. So basically the way we do the annotation is that we take advantage of machine learning tools. And in particular, this was early days where we did that, but it was quite informative as a framework to do it. And in this case, we just train a simple logistic regression model using the in vivo data, which is our reference. And then we projected the data into the in vitro data, and in this case, the organoids. And that gives us the identity of a cell, but also it gives us a probability on how good these projections are. And for example, from this, we could see that the secretory populations that are here in red, actually, they are still differentiating. They are not fully differentiated because the probability was not as high for this population. And once we have this, then we can also compare the pathways with the transcription factors. We also did, and we also see this Wnt and not this balance between the ciliated and the secretory, meaning that they are in the process of being differentiated, but not fully differentiated, probably because we also don't have the supporting stromal cells. There are only epithelial cells present. Okay, so then we use this model to validate and to inform if we can do lineage-specific models by altering the Wnt and not signals that recapitulate the different environments. And to do that, we do single cell, but also staining, and the staining is quite clear. So in the presence of hormones, we also add notch inhibitors, and these recapitulated the luminal microenvironment as predicted by us. And actually, you can see a lot of ciliated populations, as you expect in the lumen, and very few secretory populations. And if you move into the glands, then here you produce them by adding Wnt inhibitors in the presence of hormones, and then you have a more glandular-like phenotype. You have a lot of secretions. And also with the single cell data, you have a new population popping up, and I will come back on this. So what single cell transcriptomics data allow us is to quantify what is happening with these signals. We can see the difference on efficiency, which was also quite visually evident, but also we can determine the accuracy of our perturbations and the populations that are emerging in these perturbations. And as I showed you before, there is a new population of secretory cells emerging, and we show in the presence of Wnt inhibitor, and we show that those are not only more secretory cells, but also they express more secretory molecules. And likewise, we can dig into the mechanisms, and in this particular case, we were interested in the interactions between hormones and pathways. So with all of these, then, we can create models of the endometrium and how the different cell types that are present, the different signals, and how they push the identity of the cells, which is very valuable for all the in vitro work that then we can do. And indeed, in the lab, we have been improving all these references by looking at more phases of the regenerating endometrium, and then I won't go into detail, but then you can see a lot of colors, which means a lot of cell states, which we have also validated where especially located they are and what niches are occupying, making a better model, which really indicates the different populations, what are the niches that they are locating, and the specific time where they are appearing. And the reason this is important is because then we can start looking at the specific signals, not only between the lumen and the glands, but really the gradients that are present in the tissue, including the basal layer, which is where the stem cell progenitors are, and we indeed did the signals that are required to maintain this niche. And what we are very excited about this is, of course, to understand what happens in disease, but also to start looking into better more complex in vitro models. And for this, we are collaborating with Linda Griffith team. And indeed, we have implemented her beautiful co-cultures of epithelial and fibroblast cells, and we have profiled with the single-cell transcriptomics, we're showing that we have more complex and better glands being produced. But also, we are working with her to make, and she's with her bioengineering tools, improving these, getting informed about all the gradients that we see in the tissue and making a true mucosal barrier interface where we can also start complexity on the cell types and studying, including the immune cells and how they are contributing to the tissue. And please see her poster on Thursday on 187 to know more about this model. Okay, so now I am going to change gears a bit, and I'm going to talk about the endometrium during pregnancy, but the framework is going to be quite similar. So in this case, what we are interested is in what happens in all the transformation that happens in the early pregnancy in humans, which again is a black box, because I'm not talking about the very, very early. I'm talking about between 3 and 7 post-conceptional weeks, which is, again, we know almost nothing about what's happening, only some tissue slides. And again, this is different from mouse, so we really need to get information before we do any in vitro model of what's happening in vivo. And in this case, it's a very fascinating scenario as well. So in this case, we have the endometrium, which is in close contact with the placenta. And the placenta is a tissue that is built by the embryo. So really, this is a unique scenario where cells from two different individuals, the fetus and the mother, coexist together to do a function. And what happens here is also quite fascinating. So we have the trophoblasts in red, which are the building blocks of the placenta. So they are the epithelial, they are called trophoblasts, but they are epithelial-like cells of the placenta. And what happens is that these fetal cells, they detach from the placenta and then invade the maternal uterus, where they intermingle with maternal cells. And this invasion needs to happen because the arteries need to transform to enable sufficient nutrition to the embryo. And also they need to be controlled because they need to fuse into the giant cells at the border of the myometrium that do not longer invade. Otherwise, we will have the fetus invading the mom all the time, and that will be problematic, as you can imagine. And this process was the one that my PhD student, Narjit Nanan, asked herself when she started her PhD and was published recently in Nature. So the questions that Anna asked is what trophoblast invasion is fundamental for pregnancy in humans, but how is this trophoblast invasion happening? So first, she asked what are the mechanisms that trigger and stop invasion. Shallow invasion will lead to deficient arterial transformation and pregnancy complications, and is the major cause of pregnancy complications such as preeclampsia, preterm birth, and all of this. And then the contrast, the deep invasion, as you can imagine, will cause the placenta touch so deeply into the uterus then you can have uterine rupture, and that's what happens when the tissue implants into another tissue, such as the fallopian tube. So then there is some signals in the endometrium that are not present in other tissues that are controlling this invasion. And the other question that she asked is, what are the molecular mechanisms that regulate the identity of these cells as they encounter the different niches of the endometrium? And this is again crucial for this arterial remodeling. And responding these questions, on one hand, will allow us to learn about the fundamental process for how successful placentation work, and also will lay the foundation of how can we build better post-implantation models in vitro. So profiling this deep invasion during the first trimester was quite challenging. During my postdoc, I focused on the early invasion, but getting access to the full and in-depth invasion in humans is more challenging because this tissue is not that much accessible. And indeed, the only tissue where you can explore that is in historical collections of first trimester hysterectomies of uterine implantation sites. And then I will tell you how we managed to make a use of these tissues that are more than 30 years old and that were collected by the pathologist Professor Ashley Moffat in Cambridge. So before I go into this, let me tell you a bit about what we know about the tissue and how also then afterwards how our study transformed what we know about this tissue. So as I told you, this is two different tissues that coexist together, and you can see the different shapes and different morphologies. We have the placenta, which is fetal, and the endometrium, which during pregnancy is called decidua, that is maternal. And then you can see how the placenta is invading into the endometrium, and you can see these. You can see the cells really. I think this is not working, but you can see the cells are really going into the tissue. And then also you can see what I told you before, this transformation of the arteries, and you can see these big cells, which are the fetal trophoblasts, transforming the cells, and this finally, towards the end of the invasion, the cells fusing into these giant cells that do not longer invade. And what also we can see in this tissue is that we have maternal immune cells, which are the small dots that intermingle with the fetal trophoblast. So there is a dialogue, but we don't know what are the molecules that are exchanged between these cells. So the way we profiled the tissue again was taking advantage of the single-cell genomics, both transcriptomics and open chromatin. And we did a strategy where we have the frozen tissue blocks, and then we take slides and we profile consecutive slides for this nuclei extraction and single-cell profiling and spatial profiling. And what we can do here as well is to look at the heterogeneity of the trophoblast. Again, it's cell represented by a dot. And towards the yellow, what you can see is different colors, and those are the invading trophoblasts, showing how our study really showed different identities that were previously unknown, and those are represented in different colors. And what single cell allows us is to order cells into a pseudo-time. And to do that, basically, the principle is to order cells based on similarities on their transcriptome. And basically, we can see that towards the uterus, we have the invasion and how the progenitors give rise to the different identities. Towards the left is the vascular trophoblast, or towards the right is the one that invade and finally fuse. And so far, you need to believe me that with the markers, we have been able to assign identity of cells, but I'm going to show you that this is the case by making use of the spatial transcriptomics, in which I told you that there are these two differentiation pathways, one that is the invasion pathway, and as you can see, is that when we locate into the tissue, you can see the different colors representing the different cell states and how they are really showing an invasion front, And on the other hand, we have the trophoblasts, which are, you can see these plaques forms in the arteries, which are these vascular trophoblasts that we also identify, as you can see, present, and really indicating that what we see in the single cell data is what you also see in the tissue. And that is great, so we have new heterogeneity, but the reason this is important is that now we can know what are the signals that are required for these trophoblasts to differentiate. And to look for those signals, what we look is at the transcription factors, and we look both at the expression and their activity by looking at this open chromatin and where are these genes being bound. And the expression is, as you can see here in this plot, shown by the color and the different symbols represent if they are active. And with all of these, what we can do, it's a model. And what are the different pathways that are being active as cells differentiate and also encounter different environments within the endometrium. And this really matches as well with our predictions of the endometrium niche in the other work that I showed you before. For example, this Wnt and Notch disbalance will also affect the identity of the trophoblasts that invade. But we want to know more about these signals and really knowing more about the specific ligand receptor interactions that led to either trophoblast invasion or the vascular transformation. And for simplicity in this talk, I will tell you about the trophoblast invasion and how we do that. So for that, again, we took advantage of ligand receptor interactions. And here again, the way to see this plot is that we have the receptors on the left and then the ligands on the right. And then the trophoblasts are expressing the receptors, and then the ligands are expressed by cells in the endometrium. And one of the things that we see is that some of those cells that are giving the signals, or the majority, are immune cells within the endometrium. And in this is tissue that is enriched on immune cells in comparison to other reproductive tissues. And in particular, the macrophages were giving a lot of these signals, which we then further validated using special methods. And you can see, for example, in blue, here are the trophoblasts, which are sitting close to the maternal immune cells. And then you can see the cytokines and receptors that they express, making a model by which the macrophages secrete molecules that enable these trophoblasts to invade. And of course, this is a prediction, and then we wanted to use the in vitro models to validate the effect of the cytokines, but before we need to benchmark what we have on the dish, which we don't know yet. And first, let me introduce a bit of the different models that exist for this. So one is primary trophoblast organoids, which form 3D structures. The problem is that they are primary derived, so you depend on first trimester samples. And the others are trophoblast stem cells, which are more scalable because they are established, they are derived from established cell lines. But what is cool about both systems is that if you change the media, then you can see this invasion, this epithelial mesenchymal transition happening, and this invasion happening. So we wanted to know which model is better recapitulating the invasion, which is what we wanted to validate in this case. And for this, we used the same exercise. The model was a bit different, but the idea is the same, is to train a model with all the in vivo data that we have, and then project this data into the in vitro data, so then we can get an accurate quantitative measurement of what we are having in the dish. And you can see a lot of colors, meaning that the cells are well represented, and also the arrows showing that the invasion is recapitulated in vitro models. But we can also see that these vascular and giant cells that I told you before and that we see in the in vivo data are actually not present in the in vitro organoids, so there is a lot of potential, and I will go back to this, on making these in vitro models more mature and better by learning on our in vivo data. But for now, we wanted to study the invasion trajectory and how the cytokines from the immune cells influence them. And because we wanted to test a lot of cytokines, we went for the 2D model that was more scalable. And basically, what we did here was to grow the cells and then to add the cytokine, and again have a readout that was more just imaging, but also a single cell to know what was happening. And we could see here that the different cell stages they invade, they also start expressing genes that are important for motility, really showing how the cytokines secreted by macrophages are enhancing this invasion of the trophoblast. So altogether, we have generated a model on how this invasion is happening. So we have all the pathways and the stepwise pathways that we need to induce to create these populations. And also we have identified signals from the environment, and for this presentation I show you from the maternal immune cells that we need for these cells to appear. And we are super excited about this work, on one hand because we have decoded this black box of pregnancy, so it allows us to get fundamental understanding of pregnancy complications, but even better to get a blueprint for designing better in vitro models, which in this case, of course, they are relevant, for example, for toxicology screenings. And this is something that we are doing in the lab. So now, as I told you before, we have organoids or 2D systems which differentiate only up to a bit. But now we are putting combinations of transcription factors to drive that are informed by the in vivo data to drive the differentiation of the final stage into the different lineages. And we also want to see how the extrinsic factors, so the factors from the endometrium, allow this invasion, and for this we are hoping to use the more complex systems. And with this, I have focused today on our work on reproductive tissues because I thought, and on the uterus, because I thought it was a clear example on how you can do this for different systems. But of course, you can use our approach or similar approaches to investigate what you have in the dishes by using single-cell genomics and comparing with all this rich data that you have in vivo. And indeed, one of the things that we do in the lab, because we're interested in the immune cells, in how can we improve the IPS differentiation and differentiate different cell lineages, and including we publish a protocol on how informed by the atlases of the bone marrow and the different developmental organs, we can produce dendritic cells in a dish. And of course, we are also interested in starting to put these cells in different microenvironments and study the cell-cell communication. So to finalize, we need computational tools and better machine learning tools and AI to do this in vivo and in vitro comparison in a very systematic way. And one of the things that we are working, also in collaboration with Mol of Yannick, who is a new PI at the Sanger Institute, is in how can we not only integrate in vivo and in vitro data and give an uncertainty, but also to learn about these comparisons and to know what are the pathways that we need to supply to make it more similar to the in vivo data. And in a way, for this, what we are looking is into learning interpretable embeddings that allow us to map the gene expression to specific gene programs, and I'm happy to talk more about this. And my favorite slide, acknowledgement. All of this has been possible thanks to my amazing team in working in the endometrium. Was mainly led by Luz Garcia Alonso and Louis Francois, and also the new data generated also with Luz and Magda Marekova. And then the placenta work mainly led by Kevin Trulé and Arjun Nanan and Frederick Wong, and also our collaborators